So when we say drug discovery, okay, we are looking for some of the steps involved in drug discovery, right? So the first step is target identification. The second is lead, identifying the hits, then to lead, and then lead optimization. So most of the computational work that we can do is in these three steps, right? And again, drug discovery is a long process. It takes about 5 to 10 years, and it costs about $2 billion, right? So when you have identified the targets like we have done before, or nowadays you can identify novel targets also using genomic studies, right? So anyway, when you identify the target, the next thing is to identify the hit molecule. So these are some of the method that you can use, which is faster than molecular docking. Okay, so you can do 2D similarity search, you can do pharmacophore screening, and as well as shape-based screening. The results that you get from these methods, okay, they have to be evaluated using molecular docking. So they will be just looking for similarity, they will be just looking for pharmacophore based similarity as well as shape based similarity. Okay, but to see if they can fit inside the pocket of your target molecule, you have to do molecular docking. So molecular docking plays a very important role. Okay, so you can also skip all this if you have a high computational power in your lab, and instead you can go for molecular docking with virtual screening. Okay, this is more rigorous than this three steps or three methods. Okay, but it will consume more computational power, right? So once you have got the hits, okay, the hits can become leads. When this hits will become leads, when you do this adimity analysis, okay, hits that satisfy initial adimity properties and demonstrate the desired biological activity. So whatever we have talked in the previous slide, okay, those properties should be present in the hits. If it is there, then you can call them as the lead molecule. So when you have the lead molecule, you can optimize the lead molecule. What do you mean by optimization? You can enhance the efficacy, selectivity, and pharmacokinetic profiles of the lead compounds. Okay, you can use various methods. Okay, one of the method is by using QSR. You can use free energy perturbation method. Okay, many more are there. Okay, scaffold hopping kind of methods are also there. All right, so these are the methods that you can use to optimize the lead molecule. Once you have the lead molecule optimized, you can test this, or you can do preclinical trials. So in the preclinical, you will be testing an animal model, and you can, once you have the positive results from the preclinical trials, you can go for clinical trials or clinical in the you can try those uh, your potential drug molecule in the human population. So if you have positive results, you will have to go for approval. So once you have approved, then it will go to the market, right? So this is in a nutshell. Okay, so this is the workflow or the pipeline for drug discovery, and these are some of the popular methods that we use. Now you have this AI based everything. Okay, AI based 2D, AI based pharmacophore, AI based self screening, AI based molecular docking, everything you can integrate with AI nowadays. Anyways, we are not going to talk about it. So, I mean, the same thing. So, these are the experimental method for target identification. In the case of computational method, these are the some of the methods. Okay, and for hit identification, these are the experimental methods, and these are the computational, as well as for lead optimization. These are some of the methods. But you see, you cannot do this uh, computer aided drug designing methods alone, or experimental method alone. So the computational method complements the experimental method. Okay, when you combine these two, you can do it faster, as well as you can do it more scientifically. Okay, I mean, yeah, so it complements each other, right? So those who are only into computational method, whatever the model or the results that you have uh, generated using computational uh, techniques, it has to be validated by experimental studies. Okay, so if you want a good research coming out of your computational method, then it's always better to collaborate with the experimental lab. Okay, so we can again categorize computational computer-aided drug discovery or computer-aided drug design method into two. Okay, when you are including the information about the target, then it is called the structure-based drug design. Okay, so if you are have, uh, using the information about this target drug target molecule, okay, so it is called as the structure based drug design. So some of the methods are this structure prediction methods, molecular docking, receptor based pharmacophore screening, molecular dynamics simulation, etc. Okay, so if you are not using the information about the target, then it is called as the ligand-based drug design. Okay, so 3D QSR studies, ligand-based pharmacophore modeling, cell-based screening, etc. are some of the methods included in the or categorized into ligand-based drug design methods. Right, these are some of the success stories. I will share the paper where I have taken this from. Okay, so using docking homology modeling, this drug was no FDA approved. Okay, and it is used for cancer. Okay, lung cancer. So similarly, we have molecular dynamic, this docking and molecular dynamic simulation for this particular drug and some of the few drugs also.